Have you ever thought of using bacteria as a memory card? Well, Yim, along with other researchers, have thought about it, and they have managed to store digital data in the DNA of living bacteria. They published this article in Nature Chemical Biology in January 11th, 2021, and we're going to talk about what they did, how they did it, and if we could use it in our daily lives. But why use bacterial DNA to store digital information? It has a high density storage capacity. With 1015 bytes per gram, the system allows to save information of up to 72 bits. It has a great long-term stability because its integrity is maintained throughout multiple cell divisions. And also, it has a low risk of obsolescence in the future. Technically, the binary system has already been used to codify in vitro synthesized DNA, but they did not synthesize DNA in vitro, which is the technique usually used. So, what did they do? They used electrical signals to transfer digital data from a computer to the genome of living cells. And I guess you are now wondering, well, but how did they do it? The system was based on an adaptation of the CRISPR-Cas9 gene editing tool in response to an electrical signal. The bacteria synthesized a high number of copies of a plasmid. Plasmids are free circular DNA molecules separated from chromosomes, which, among other functions, play an important role in the development of resistance to antibiotics. CRISPR-Cas9 then cut out this excess plasmid molecule and placed the fragments in specific regions of the bacterial DNA. This action would encode the value 1 of the binary code with the genetic material. Instead, in the absence of electrical signal and therefore few plasmids, CRISPR-Cas9 would insert a generic genome sequence corresponding to value 0. In other words, scientists managed to store a binary digit or a bit of information in DNA. And this is amazing. However, the investigation did not end at this point, since they found a way to increase the storage capacity to 72 bits, a fact that allowed them to encode the message "Hello World" in the Escherichia coli genome. To read it, it was enough to sequence the DNA of the bacteria. It should be noted that the stability of the stored data was quite high. After 80 cycles of cell divisions, and despite the mutations acquired during the process, the message was not noticeably altered and could be decoded with 19% of accuracy. Likewise, even when exposed to a natural environment, such as soil populated by other bacteria, the bacteria preserved its integrity of the encoded information, while free DNA degraded in less than 6 days. This finding is promising, but there is still a long way to go before living systems can compete with current data storage systems, and therefore we could use them in our daily lives. Thanks for watching. If you want to know more, check the links in the description. Bye!